Hello. Right now we're going to talk about the process of replication, pretty much the process on how the DNA code is copied to provide an exact copy of itself. This takes only a few hours to copy all of the DNA, which is quite impressive. And you only really get a few errors, about one error per billion nucleotides. There's about more than a dozen enzymes and other proteins that help participate in this process. But pretty much, replication is the process of how the DNA code is copied. We already learned that it occurred in interphase of the cell cycle and interphase 1 in meiosis. If you remember, interphase 2 is just preparation. And it is where new strands of DNA are made from a supply of nucleotides that float freely in the nucleus. So this process of replication occurs in the cell's nucleus. This is just showing you what happens is the DNA has to unwind, the strands separate, and then free bases come in to actually complement the base pairing. So, step 1, okay? An enzyme called helicase is going to break the hydrogen bonds between the base pairs. Okay? That is going to cause the helix to unzip and expose the bases. And this could occur at many different places within the DNA molecule at the same time. It actually occurs at parts called replication forks. Now, the free-floating nucleotides will pair with the exposed bases, and that is going to be done by the enzyme DNA polymerase. Now, we only can add nucleotides to the free 3' end, never to the 5' end. All right, so it's always going to make the DNA or elongate the DNA in the 5' to 3' direction. And the rate of elongation is quite impressive. It's about 500 nucleotides per second in a bacteria and about 50 nucleotides per second in a human cells. Okay, and that again is just a little fun fact for you. Okay, so again, we have to separate the strands, and then once the bases are exposed, we then can pair up the DNA. Okay, so again, bonds break, the strands separate, and the free base pairs do add in together. So the point at which replication occurs is called the replication forks. And again, this can be multiple parts of the DNA molecule at the same time. And what we get are called these little replication bubbles. And this is just showing you again how the strands are anti-parallel to each other. So, DNA polymerase adds nucleotides only to the 3' end of the strand. So the daughter DNA strand, the new strand, only can grow in the 5', 3' direction. And again, just showing you how they are numbered. You can see that each of the carbons are numbered. Okay, so we have two types of strands. We have a leading strand and we do have a lagging strand. The leading strand, okay, is the DNA strand that is made in the 5', 3' direction. It is going to form a continuous strand. Now, the DNA strand that's made in the 3', 5' direction makes what are called Okazaki fragments. So they need to have something else or an enzyme called DNA ligase to join the fragments because it is going in the opposite direction. And every time the DNA unwinds, it exposes more bases, and those are what causes the fragments to be made. So just to kind of show you that as the picture, okay, this can be made continuously. This gets made this way. When this unwinds more, we make a little fragment, and unwinds more, makes a fragment, and those Okazaki fragments do need to be joined together by DNA ligase. And again, it's just showing very continuous, and then fragment, fragment, fragment. And as this unwind more, it'll be another fragment, where this one can just keep going. And we're going to do nice activities to help in explaining all this. So if you have any questions, just let me know, but we will review this quite a bit. And that is the process of replication. So if you have any questions, let me know. If not, I'll see you tomorrow. Great, thanks.